Hello, everyone, and welcome to our third session of SalivaDirect Inc.'s annual conference. We're very excited to have you here. This is our fourth annual conference, but our first virtual one, and very pleased that there's so many individuals joining us from all around the world, both live as well as watching this recording later on. It's my pleasure to welcome our speakers here for this special session. We're going to be doing an Ignite Spotlight presentation, which is giving our presenters from the poster session that will immediately follow this, a few of them the opportunity to share their information live, to be able to communicate what they've been working on, and have a brief opportunity to showcase what their work is all about. First up, we have Ravneet Grewal, who is joining us from India, and she's presenting on Mucin in Saliva, a promising non-invasive tool for tobacco-related oral health. Ravneet, over to you.
Thanks, Brittany, for introducing me. Today, I will be starting this conversation with the word crisis. We all know that in crisis, each little effort counts. But the important thing is not to wait until you are in a crisis to come up with a crisis plan. And this is the rationale behind this study. Here, the crisis is oral health issue, which stems from long-term use of tobacco and high doses of tobacco. And there are many, ranging from potential malignant disorders to oral cancer. Now, the question is why I'm labeling this a crisis when we have multiple other health risks in India. For this, you need to look at few statistics. Okay, so I was discussing the reason behind taking oral health issues as a crisis in India when we have multiple other health issues. For this, you need to look at few statistics. India has reported the highest burden of oral diseases. Alarmingly, India has the highest number of oral cancer worldwide. And 90% of these cancers, they are associated with tobacco use, long-term tobacco use with high amount. There is another report, National Health Family Survey in India, which asserted the prevalence of tobacco among men compared to women. In India, every fifth individual is reported to use smokeless tobacco, while every 10th adult smokes tobacco. In lower income group, the most common tobacco products are bidi, tobacco powder, and gutka. These three tobacco products cost very little, and they are common among this lower income group. Now, this is the population which is vulnerable to tobacco-associated oral cancer. Now, the matter of concern is that these individuals, they are mostly diagnosed at late stage, and it adds a burden of treatment cost to them when they cannot afford the treatment. Though Indian government is taking preventive measures, but all these efforts, they fail to address the rising epidemic. At this time, I remember a quote that an ounce on prevention is worth a pound on cure. But in this case, when the preventive measures, they are failing to deliver, then I would say an ounce on prognostication is worth a pound on prevention. With this in mind, I started looking at oral defense network, and I came across a list of defense molecules. Out of these molecules, there is one particular protein which caught my attention, and this protein is a mucin molecule. When you look at this molecule, you will see that it is very fascinating with conjugation of proteins and carbohydrates. If you look at the residues of the polypeptide backbone, then proline, serine, and threonine, these are the three amino acids which are potential site for glycosylation. When this domain undergo glycosylation, a dense array of O scaffold is formed, which and this state is essential for the physiological function of mucin, which also include oral defense. Then, Then these mucins, if you look at the glycan structure of these mucins, you will find that they are often terminated with fucose and sialic acid. These two molecules, they are crucial for fundamental physiological functions that include pathogen recognition, cell adhesion, and also immune response. So with all this information, I got curious to understand what would happen to mucin terminal glycosylation status if the oral cavity is exposed to high doses of tobacco for a long time. So for this, we collected saliva samples from tobacco habituates, and we estimated mucin levels in their saliva. And when we got the results, we were thrilled because we found that these mucin levels, they were reduced significantly in smokers who had been using high doses of BDs. For BD is a non-filtered cigarette which is commonly used and it costs very little. That's the reason it is common among low-income group, which constitute the major population of India. So these smokers, they reported significant reduced level of mucin compared to healthy individuals. When we extended the study to gutka consumers and the tobacco powder consumers, these are the smokeless form of tobacco, or you can say they are usually chewable form. Analysis in gutka consumers and tobacco powder consumers, we found that again a negative correlation between these two parameters. So these results, they clearly defined that oral mucosa get compromised when it is exposed to high doses of tobacco for a long duration. Now, these results really encouraged us, and the second step we took was to analyze these terminal residues, which are terminal residues of mucin. So when we estimated sialic acid content of mucin, we found that mucin silylation is enhanced in all tobacco habituates that include both smokers and smokeless tobacco consumers. And when we try to estimate the fucose content in saliva, we saw the same trend. So these results made it clear that terminal glycosylation status that undergo aberration on chronic abuse of tobacco. And these results also highlighted that it is the duration of tobacco use that actually had impact on the loss of the mucosal integrity in tobacco habituates. In the end, I'll conclude with a key message that this aberration, terminal glycosylation of secretory mucin in saliva could be used to trace the molecular alterations which are associated with tobacco pathogenesis in oral cavity. I think I'm over. Thank you.
Thank you so much for your presentation. Looking forward to the Q&A section. Up next, we're going to hear a presentation from Lavinia Alberi Auber, who's joining us from VitalizeDx, which has bases in Switzerland and Italy. And she's going to be presenting on the association between salivary microbiological parameters, oral health, and eating habits in young athletes.
Thank you for having me today. And I would like to present this work, which was published on the Journal of Athletes Nutrition. It's a work that has been done with the University of Chieti in Italy, dedicated to oral health in athletics. As we have heard, oral condition are widespread, and dental and periodontal diseases are the most common non-communicable disease worldwide. And those conditions are really just the top of the iceberg. Chronic oral dysbiosis causes immense damage, such as chronic inflammation that really affects both the adults, the young, the adults, but also the elderly. In particularly, many of the oral pathogens do have an effect on the cardiovascular system, and those are just some of them causing chronic inflammation. We focused on athletes' oral health because athletes are particularly susceptible to oral dysbiosis due to their increased cardiometabolic risk, a diet which is very rich in sugars, and also the use of mouth guards. And so we posited whether athletes were at risk of oral dysbiosis and sequelae of oral dysbiosis, such as myocardial infarction. This has been also the subject of a grant that we got, which is called OraCardio. The study that we conducted has examined four type of sports in young adults. They were practicing swimming, soccer, water polo, skiing, and basically we counted 56 variables, including dental parameters like DMT, so dental missing filled teeth, active caries, plaque index, gingival index, dental stain, and erosion, the effect of oral care on those, eating habits, functional food, snacks, and composition of the meal, with the objective of finding dependency between those variables. So we started with looking at the load, the dysbiotic load, and we found effectively that Streptococcus mutans was positively correlated with the plaque index, and the dental stain were actually correlated with the gingival index. If we look at the oral care, we also observed that the brushing 3 times a day is better than brushing twice a day, because by brushing twice a day, you have a higher DMFT. Looking at the effects of the number of times you're eating, it doesn't really make much of a difference, but what you're eating does make a lot of a difference. So if you're looking at functional foods that athletes eat all the time, especially the one with the content of chocolate and milk, you see that there is a strong correlation between the PI and GI and the dental stain and the dental erosion with the snacks and the chocolate. And there was found actually an inverse correlation between active caries and dental stains. On the other hand, the snack and fruit were also positively correlated with dental erosion and dental stains. And if we look actually at the breakfast looks very similar to the snacks. We have a strong correlation with the morning coffee with dental erosion, morning milk would actually associate with the plaque index, and morning biscuit with dental stains. So a lot of content of milk does associate with those dysbiotic variables of oral health. In terms of the lunch seems not to, the content of the lunch doesn't seem to much associate with those disease variables, neither dinner. When we looked at the 4 different groups, we saw actually that water polo was distinguishing itself much more than the other sport, following skiing, swimming, and soccer. And then we looked at those variables in aggregate, and we saw that aggregate disease score, accounting for the DMFT, PI, GI, the dental erosion, dental stain, was the highest in water polo. We also looked at the water polo skier were actually were brushing twice per day less than the others. We're actually brushing more, but still the water polo player had a very high Streptococcus mutans lactobacillus species load. We also observed that what was emerging is that they had a very high intake of energy snacks and chocolate that we saw that were associated with the disease biomarker. If we also looked at the pH, we understood that water polo do have actually a decrease in their pH after the game, and this decrease is probably one of the reasons why they do have a more dysbiotic environment. We then looked at the interaction between the energy and the stackness chocolate and the load of Streptococcus mutans and Lactobacillus species, and we find that there was a very, very strong association between the two in the water polo group. Then just plotting all those variables, we observed that when we were looking at the Streptococcus mutans, the load are very high. We do have also this association with the brushing twice per day, which apparently is not optimal. And following the water polo, we found also the skiers, which were brushing twice a day, and also had higher Streptococcus mutans and Lactobacillus species. Looking at finding dependency, we ran a logistic regression, and we found that energy snacks and chocolate were effectively increasing the likelihood of the appearance of Streptococcus mutant Lactobacillus species 6 times, and Streptococcus mutant Lactobacillus species were increasing 13 times the risk of the aggregate disease. If we then do the association between energy snacks and chocolate, we saw that energy snacks and chocolate were increasing 12 times the risk of the aggregate disease score. So it seems that milk products in athletes are creating this dysbiotic environment, which is very permissive to pathogenic species that could cause cardiometabolic and cardiovascular issues. So this paper was published at the beginning of this year, and this is exactly what we say. Water polo are more subjective to suboptimal oral health. We do have this very high load in Streptococcus mutans and Lactobacillus acidophilus rate, and skiing overall has the worst dental practice, maybe because they don't have the time to brush. We then investigated whether water polo was more subjective to this dysbiotic environment due to the mouth guard. And effectively, in a pilot study, we observed that Streptococcus mutans does effectively associate with the total load, and also the presence of Lactobacillus acidophilus. So the two bacteria are symbiotic, and there is also an association with Candida albicans. And so I would say that oral health needs to be taken into consideration in the global health of athletes. We know that those athletes can be at risk, and sometimes they drop dead, and we don't know why. Maybe looking into the oral compartment would bring some solution. Of course, this is very easy to study, but of course also the implementation of our care specific products such as gel with nitrate based food, and maybe some less sugar would be optimal. Thank you.
Thank you for your presentation. I'm already thinking of a few folks that I'm going to need to send this particular slide deck to. Up next, we have our third and final Ignite presenter, and that's going to be Michael Hogan. And he is joining us from GenTegra, LLC. And the presentation he's going to offer is ambient temperature stabilization of nucleic acids in saliva by a combination of nuclease inhibition and the inhibition of microbial overgrowth.
As the previous talk said, and all of you know better than I do perhaps, saliva is a very important source of sampling for any number of things, from human genetics to cancer risk, drugs of abuse, and so forth. It works great, actually, as long as you're analyzing it relatively quickly, which most people do. That is within a day or a few hours. What we're specifically focused on is extending the power of saliva collection to support at home, at school, institutional collection, where in fact it won't be possible to do the analyses that are interesting and important on site or point of collection, but you're going to have to ship it, and which may take several days, refrigeration, and so on. What we want to do is and we'll be talking about today, is the ability to develop a new technology that very inexpensively can support this kind of at-home collection. I'm not advancing. There I go. It's a little slow. So how to make saliva collection more robust for at home. So the untreated saliva is very well known and useful. And in fact, if you're going to use it quickly, is very well suited. There are methods that are currently available to stabilize saliva, particularly nucleic acid in it for obvious reasons, for ambient temperature shipping. But they generally are kind of a big hammer, so to speak. They involve adding very large amounts of a solvent like alcohol, 50% actually, diluting the sample 50-50. Things like 0.2 to 0.4 molar guanidinium hydrochloride, very good at stabilizing nucleic acids, inhibiting microbial growth and so forth, but you've got to get rid of that as well. And then those additives very often make it very difficult to analyze salivary proteins and small molecules because you've got to get rid of them. So the question is there a more surgical or precise way to modify saliva with minimal intervention to inhibit the things that should be inhibited, if in fact you need to hold the sample at room temperature for a week or two while you're shipping it, storing it, and preparing to analyze it at some distant site? First of all, why do you do all of that stuff? Why do we put saliva or anything else in a refrigerator for that matter? And it's for obvious reasons. For those of us interested in nucleic acid, you would want to inhibit, DNase and RNases in them, general base catalyzed hydrolysis, even if there aren't any enzymes, oxidation. Proteins are very sensitive and small molecules to oxidation. And more recently, kind of the elephant in the room, everybody knows there's a salivary microbiome. That microbiome, if left to its own devices over several days, can incur substantial outgrowth and produce what might have been a small microbial load, can bloom into something that's very large, to produce direct and indirect effects on the nucleic acids, the proteins, the small molecules, throw out nucleases, produce an enormous amount of undesired, commensal DNA in these microbes. And frankly, all the things we're interested in are just food. And so you can have catabolic loss as well. So what we've done is as an extension of what we've done for some time, based on DARPA and NIH funding, National Institute of Health funding in the US over several years, we've been focusing on, in other contexts, developing small molecules that can be delivered at millimolar concentrations to avoid dilution and adding large amounts of solutes and solvents that have to be removed before you can do anything with your sample. And we've applied that here. And so the basic performance requirements that we've set for ourselves, that I think we're pretty close to having achieved, and we'll talk about: make an additive that's stable, small, and can be applied as a little film in the bottom of a tube, dried, a coating, so that you could directly or indirectly apply saliva into the tube and mix it, and there you go. And you've only really diluted the sample by 1% or so, and you're not trying to get rid of a lot of stuff. And that we want it to be such that, having been put in there subsequent to 14 days, we set a standard for ourselves that success is only going to be defined as greater than or equal to 14 days of room temperature ambient stabilization to inhibit nucleases, oxidation, and microbial growth. Those three things are, I think, not just important with respect to nucleic acid analytes in saliva, but proteins and blood drugs of abuse and so forth. And this microbial growth thing, I think, is not only, of course, important if you're worried about bloom affecting the quality of your analytes, but if you're actually interested in the microbiome, I think the fact is that after something's been sitting for a week or two at ambient temperature, the microbiome that you measure at that point has very little to do with what you started with. We're going to talk about two things today. This concoction is called STM+, and sometimes I'll just shorten it and call it STM. It does two things that are important to us, and I think generally important: inhibit microbial growth in saliva at room temperature for a very long time, at least 2 weeks. And we would like it, in the course of doing that, to stabilize any number of things, but we will focus today on the nucleic acids, and particularly human nucleic acid in the saliva samples, and using a very well-known marker gene, GAPDH, as the sort of surrogate for the rest of the genome, and analyzing by real-time PCR, qPCR. First, we'll talk about the inhibition of microbial growth. First, we'll show we set up a small study, half dozen. I'll show the outcome of a half dozen volunteers, and basically just healthy people collect saliva at time equals 0, take a milliliter or so of the saliva and either place it in a tube that has had this STM plus dried within it, or a match tube that has nothing, and just incubate it over time and see what happens. First, we're going to show you what happens just by simple images, and then we'll show you a little bit more quantitative data as to the bloom of microbial growth. And the methods I won't go through in detail, but basically we're using ordinary, blood culture, which is kind of the general way to look for total microbial growth, particularly bacteria. And when we get down to the nucleic acid part of it, some of this, an identical aliquot of the sample that had been submitted to plate culture had the DNA extracted out of it by using absolutely ordinary, standard of the art DNA extraction, followed by real-time PCR. First, here's just a few quick visuals. What you're looking at here is, from several donors, after day 0, meaning as soon as the saliva was collected, it was plated. Day 2, it had been sitting around at room temperature for 2 days, then plated day 7 and day 14, and so on, and the untreated saliva on the top. And the STM plus treated, i.e. a tube that had this dried stuff in it to which a mill of saliva had been placed. What is obvious is that, these aren't striking pictures because the blood agar makes the background kind of high, but you can see there's a lot of microbial growth on the top, which is the untreated material, and exactly identical specimens at exactly the same time points show no evidence visually for microbial growth. And then pretty much the same story for another donor, and so on. Digging a little deeper at day 7, we did a little bit more detailed plate enumeration rather than just showing you some pictures. We tried to count colonies, and the samples are listed 1 through 5, S1 through 5. And basically what you can see, they've been on saliva. STM means it's saliva to which has been placed in an STM tube, using the slang I mentioned, stored at room temperature for 7 days. And the bottom line is everybody who was untreated saliva showed massive bacterial overgrowth that could not even be counted if in fact the saliva were diluted 1:10 before plating. So lots of microbial overgrowth, okay? In the presence of STM plus, the small molecule that we're adding to induce growth stasis is doing its job. There's no detectable colony growth on these match samples that were completely overgrowing the plates. There was one interesting sample, I don't know what the answer is, but one sample, S4, apparently either had so much bacteria in it or a slightly different mix that we didn't get complete inhibition. It's still enormous inhibition of microbial growth compared to no treatment, and we're in the process. Probably if we saw it here, we'll see it other times. There may be, outliers in terms of how much ambient bacterial contamination or the mix that we may need to do a little bit better job of shutting it down. But we're doing a pretty good job here. The second thing is then kind of the molecular part of this. Again, DNA is not the end all for all molecules, but everybody is very interested in the DNA, human DNA complement of saliva. Again, the exact same study design. In this case, during the time points, we fetched 200 microliters of saliva, did a standard DNA prep, a standard qPCR analysis, using a commercial kit for GAPDH, and used that to tell us, can we detect GAPDH, and how's that changing over time? This is kind of the money shot for the whole presentation here. What you're plotting on the y-axis, we're plotting is the so-called CT values for those of you who are nucleic acid jocks. It's a measure of the quantitation of how much PCR is going on. The green bars are for specimens that have had no STM added. The orange bars are those that were placed into a tube with the dried STM film and it was dissolved. And it's pretty obvious. And the x-axis is time and days of incubation. It's pretty obvious. The first thing, it's actually for of technical importance. At day 0, where there shouldn't have been any time for anything much to have happened. I mean, saliva works fine. There's not a lot of DNA degradation in saliva over a day or so. And the fact of the matter is that indeed the two with and without the STM treatment are the same. Out to about day 2, they're still pretty similar. Again, saliva is pretty good at stabilizing DNA without any fancy additives. But by the time you get to day 7 and day 14, the apparent amount of the GLAP-DH is dropping drastically. Again, big numbers in CQ values correspond to reduction. And plotting that same data on a sort of differential plot, by the time you get to day 14, there's been about a 10 CQ value using nucleic acid talk. There's been about a 10 CQ value change in the GAP-DH signals, which as a first approximation corresponds to about 1,000x drop in the amount of intact GAP-DH after 14 days. And then the 6th CQ value after 7 days corresponds to about a 300-fold drop. So once you start to pick up a head of steam, so to speak, the fact of the matter is that untreated saliva is simply not suitable for, at least, again, it's dangerous extrapolating to all genes based upon this, but it's likely that the indication that is, in fact, as long as you want to use saliva without any fancy treatments for a day or so, that's fine. At ambient temperatures, though, by the time you get out to about day 7 or day 14, there's not only been an enormous amount of microbial overgrowth, but possibly in a related way, there's also been a big reduction in the amount of and the integrity of the nucleic acid in the sample. Just to finish up, the bottom line is that we've shown that STM Plus is doing a pretty good job of inhibiting undesired bacterial growth that may contaminate molecular testing. And if you happen to be interested in looking at the microbiome, it probably would be a good thing to have in there as well. During this inhibition of microbial growth, the rest of the stuff that's in STM Plus, the antioxidants and the nuclease inhibitors are doing their job, and at least as seen from this small sampling of GAP-DH, the DNA appears to be intact. Work in progress with collaborators is obviously extend this to other nucleic acids. Can we similarly stabilize RNA in such samples, but also protein in such samples, and small molecules such as drugs of abuse, which are sort of very sensitive to oxidation. And because we sort of developed the technology so as to be friendly to be placed into collection devices as we speak, we're in the process of collaborating with a company who sells a well-known saliva collection device to put the STM Plus in there so that the act of collecting the saliva and direct transfer into this stabilization media sort of happens in a natural way. With that, I want to thank you very much, and I'll answer any questions later when they come up.
Great. Thank you, Dr. Hogan. I appreciate that. As you noted, we're going to transition over into the live Q&A portion of today's presentation. And there are a couple of questions that have come in for all of our presenters. So if Lavinia and Raveet, you guys wouldn't mind joining us back for this. But first question is actually over to Ravneet, and that's a question about what are some next steps for the study? Where are you planning to share the diagnostic information that you all had to increase awareness and maybe help inform some health safety protocols related to smoking cessation or those kinds of steps?
Now we already have this information that terminal glycosylation of mucin molecule is disrupted. It is aberrant. So the next thing is to see the molecular basis of this aberration. And for molecular basis, we need to look at the proteins or the enzymes which regulate, which make sure that that these glycans they are properly expressed on the surface. If you give me a minute, I like to explain it a little better. If we see a normal cell, in normal cell, the cell surface molecule also have these sialic acid and fucose. But if you see a cancer cell, then these molecules, their expression is increased. And if we look at the reason behind, who is working behind the picture for this enhanced expression, then there are enzymes like CIL transferases and fucosyltransferases. So when we see this for mucin molecule, when we see the terminal glycan epitopes of mucin molecule, sialic acid and fucose, we see that they are also enhanced. Their expression is enhanced. So we need to find a connection, how this increased expression, which we see in tobacco habituates, is leading us to the progression of cancer. So for this, we need to look into the molecular basis. And for molecular basis, we need to look how these enzymes, CIL transferases or fucosyl transferases, they express, how they work. We need to look at their activities and expression to get more detailed picture.
Thank you, Dr. Grewal. Appreciate that. The next question that came through is for Dr. Alberi Auber, and that's, what's the potential for building out a panel for athletes to be able to identify markers? We've had previous presenters talking about how there's saliva-based testing for concussions in athletes, and you're obviously getting into the oral conditions and things like that. So what do you see as the future maybe for having a panel for athletes to better understand their health?
Yeah, definitely. I mean, athletes don't have a lot of time. So if we build a panel, it needs to be either on a point of care, portable, and we're developing that, or a specific nanosensor that detects a keystone pathogen, Streptococcus mutans, for example. I think host pathogen interaction, I mean, in terms of acute care, but also chronic care, will be really fundamental. And so for athletes, we need to be super fast in their detection and integration with other metrics. So this is something that we are doing currently, looking also at understanding how oral health does impact actually cardiovascular tone, in terms of really the effect on myocardial infarction. I think this is a study which is, of course, long-term, and we were interested in identifying those risk factors. And we know there is in saliva, we do have really a wealth of information that goes from immunological to vascular to also some risk biomarkers that we can catch up. So I think we are on the verge of really a revolution. Not many people understand that this is happening because, of course, we're very focused on blood, and blood is, the standard of care. But I believe starting from athletes who need repetitive testing and the ease of care and accessibility of saliva and the wealth of information is unbeatable.
Absolutely. Absolutely. Thank you for that. And a follow-up question that also came through, and you alluded to this a bit on, I think, one of the last slides of your presentation, is using the information that you've had to maybe inform next steps for maybe snacks or alternative nutrition intake or things like that, or maybe
Absolutely.
cleaning for mouth guards.
Yeah, no, so definitely milk product, milk energy bar should be avoided during endurance. And we actually are planning to make a gel which is based on nitrate, so beetroot, and we know beetroot is already taken by several athletes, and beetroot basically switches the environment to be more abiotic in the mouth, and instead of having a lot of sugar, which of course all those gel bar have, using MCT. So having really some nutrients which for the endurance athletes, which are training and enduring for 6 hours per day. I think this is an important, very, very important. And the milk has been already observed, this effect of the milk. It stays a lot in the mouth and creates this dysbiotic environment. But yes, we just need more paper, more evidence. But I think the athletes are under so much caloric pressure. They really need to intake good food and good energy sources with maybe a twist of nitrate.
Thank you. Appreciate that. Dr. Hogan, the next question that came through is actually for you. And of course, this also would apply to our other presenters. And that's, what are the goals or maybe some challenges to getting over to market, to getting STM to over that final hurdle of being more widely utilized and available for people to take advantage of in their own diagnostics or helping as an inhibitor?
Yeah, we think that we're sort of, our intention is to be sort of GenTegra on the inside, so to speak. I think there are a number of technologies out there that were developed by others for saliva collection that are, sort of, kit based and are suitable for at-home use and so forth. Our goal is to partner with one or more of the leaders in that area to arrange it such that our stabilizer is actually just built into the kit. It's and basically the user, other than the fact you can see a little pink film or spot at the bottom of a collection tube, these devices all have something that is put in your mouth, and then something wherein the saliva ends up being subsequent to collection. We want to be the guys who are in that collection tube, such that at that point one doesn't have to worry about either quickly getting into a refrigerator or but instead have it such that it could be done anywhere, and then barely just the whole thing could either be stuffed into an envelope and mailed book rate second day and so forth to a central lab. Or if you really are worried about getting the data as quickly as possible, you don't have to worry about don't have to worry about fluctuations in temperature and so forth. This microbial outgrowth thing is real, and we're not the first to see it. And so I think this idea that if you are worried about the microbiome, I'm a big fan of point of collection POC testing, as Lavinia was describing. If you're not going to do that, which I think it's a very good thing to do, but if you want to, to take advantage of centralized testing and things of that kind, I think it's very, very risky to not have some sort of a stabilizer introduced. And I don't think we're going to be selling that to home consumers. I think this is likely to be something that's built into a package with others who have testing programs that are important to them.
Yeah, absolutely. That kind of stability and being able to get test kits or vials out to more rural settings, more remote areas, lower resource communities, those kinds of things. It all adds up to the ability for us to be able to implement these kinds of technologies out into the field. Absolutely. Yes. And to say thank you to our presenters for sharing with us. It's really a joy to get to have presenters literally from 3 different continents all coming together to present on one virtual webinar.